Thanks, Betty, for the nice introduction. Hello, everyone. My name is Shen from University of Maryland, United States. First, I would like to thank the organizer for inviting me to come to join such a wonderful meeting. It's my great honor to be part of it. So today I'm going to talk about development of a specific phage endolysin against strep in a particular intracellular pathogenic environment. So here is a lysis from without by bacteriophage-encoded endolysin has been investigated as a therapeutic agent against bacterial pathogen due to their lytic activity. Here is the EM imaging showing this is a single streptococcal bacterial cells is exploding upon direct contact with exogenous additional endolysins. We're interested in strep because this strep is a gram-positive human pathogen used to colonize on the skin and the mucosal surface, causing infections from mild superficial strep throat to some severe life-threatening necrotizing fasciitis. And one of the interesting about strep is, although they are considered as extracellular pathogen, but indeed, many of them has a remarkable capacity to invade human epithelial cells, showing here. So that's back to the question. Their invasion process of the strep into the human epithelial cell actually can help them hide out from antibiotic action because of the natural barrier of the lipid bilayer of the plasma membrane. So that's encourage us to develop an alternative approach to engineer to make this antimicrobial to actually go intracellular. My first project came with engineering endolysin. So the way we develop our strategy is to develop approach to engineer endolysin by fusing them with cell-penetrating peptide. The cell-penetrating peptide, as someone already mentioned, there are a group of the short cationic sequence with a remarkable capacity to cross the membrane. One of the most famous case is called the tight peptide. And you can see there's a few arginine and lysine, which are defined as positive charged residues. And this positive charge residue actually can interact with the negative charge phospholipid head group in the membrane, where promote the translocation through the membrane, shown in this cartoon. So our goal is to use this peptide to label 3 streptococcal endolysins, which are B30, PlyC and PlyC, and then we want to set up an assay to test if this tau label actually can help them to go intracellular and clear intracellular pathogen like group A strep. The model we developed is human epithelial cells with streptococci co-culture model for evaluating the bacterial invasion. So showing here, first we grow a monolayer of the epithelial cells, and then we infect with bacteria, and wait for a short period, allow the bacteria actually internalize and become intracellular pathogen, and then we wash out the free-floating bacterial cells, just whatever left is adherent bacterial cells on the surface and the internalized bacterial cells. And then we treat the co-culture with gentamicin combined with the penicillin that are only target the bacterial cells in the extracellular environment. And after that, we adding endolysin or buffer to evaluate either if the endolysin has some intracellular killing activity by lysing the co-culture and recover the bacterial cells in the agar plate. So in this assay, it can really help us differentiate the adherent bacteria versus the truly internalized bacteria. So this should give you a quick result what the co-culture assay show us. So we engineering this endolysin with tau labeled, and then we testing your co-culture assay here. In the entire co-culture assay, we have around 10,000 bacterial actual cells actually go intracellularly. And as you see here, frankly, none of the tau labeled endolysin actually seem to help clear intracellular pathogen. But to our surprise, there's one endolysin, which turns out to the wild-type endolysin, the PlyC, show the best efficacy. So the rest of my talk actually predominantly focus on this wild-type PlyC endolysin. What is unique about it and why it can show this efficacy, whereas the rest of the other endolysin does not. And then we did investigate more formally by increasing the dose of the PlyC to see if this efficacy phenomenon is dose response. The answer is yes. We actually increasing the PlyC dose, the recovered streptococci decrease, and with the maximum dose of 50 microgram PlyC, 96% of the intracellular strep was cleared. And then we add heat denatured PlyC and also some other wild type, another endolysin to show that this phenomenon is truly contributed by the enzyme active domain of the PlyC and very specific to this endolysin. All right, so then we want to use an alternative approach to confirm this phenomenon. The method we decided to take is live cell imaging by confocal microscopy. So the beauty of the confocal is it can dissect your sample through the z-axis by focal plane, different focal planes. And then after you take a different picture of focal plane, you stack them together. That's what will give you a truly 3D structural view of the intracellular structure. Similar to our co-culture assay, the video I'm going to show you is briefly the cartoon, give you an indication. You're going to see a human epithelial cells, a nuclear stains, and also we have another staining, actin staining, which stains the cytoskeleton, indicating the position of the cells. And then we infect the cell with a strep, waiting for the strep to go intracellular into the cytoplasmic, and then we add our fluorescence labeled PlyC, the endolysin. If the rationale is if they go intracellular and if they target the strep, because they have the cell wall binding domain, usually record bind on the strep cell wall, it will light up the strep. And then I'm showing the video, showing the whole process. Over here, you show one single molecular cell, a cytoskeleton. What you're looking at here is actually go deeper, penetrating into the very nuclear localization. And then again, first, on the surface, you see some strep cell where they're sustained by our enzyme. And then when they go deeper at the perinuclear localization, you see some internalized strep also co-localized or stained by our fluorescence labeled strep. So in summary, PlyC, this endolysin seems to the first endolysin that has an inherent ability to clear intracellular strep in a dose-response manner. Also, we use a live cell imaging to confirm this phenomena. And the next question we ask is, what is unique about PlyC? Previously, our lab determined this PlyC has a one calic domain showing pink. And the unique thing is the binding cell wall binding domain actually contains eight identical subunits to form an octamer ring, shown here. If you rotate that structure by 90 degrees, so what you're looking at here, that's the cell wall binding domain. The surface point to us, and the far side is hooked with the PlyCA. I'm not going to talk about the detailed structure of PlyC because of my boss, Danny Nelson, going to present the unique structure of this enzyme on Thursday. But just bear with me for this moment. If you look at the sequence of the PlyC-B, the binding domain, the monomer sequence, you actually see a lot of several positive charges like lysine, arginine, exposed on the surface. When you generate an electrostatic surface map, and you see this the same surface that's exposed to super positive charge. And then we will also question, is this super positive charge mimic the penetrating peptide in this scenario? Logically, I think when you look at the structure, one thing you can do is make a bunch of mutants with cytodirect mutagenesis. So the way we did, again, I create a series of constructs which mutate all the positive charge exposed on those surface to the negative charge to the glutamic acid. And then again, we use a co-culture model to benchmark our PlyC and mutant activity. So what you're showing here, normally the wild type show 100%. The long story in short, every single positive charge mutant actually seems to have decreased activity. Some of them increase a little bit, some of them decrease a lot. We also, to confirm this really charge issue, we also make a neutral mutation and also random mutation to confirm there's either slightly decrease or no decrease on the intracellular activity. And that's back to the next question, whether we thought the decreased intracellular activity is because of prevention of this endolysin go intracellular, or to prevent the actual enzyme go intracellular losing its functional activity. So what we did is we have this mutants, and then we label them with fluorescence. The first thing we tested if they still bind on strep. The answer is yes and no. Most of the mutants here, they still bind to the strep, but one of them, arginine 66, seems lost the binding capacity to the strep. And then we use the same construct testing our co-culture model, and showing here that's the wild type electrophore label PlyC, saying they will be bound and internalized. It has a perinuclear, the red dots here showing the perinuclear localization. And this, all the mutants we have, actually they lost the internalization activity, indicating this is really charged, contributed by the positive charge. And then we did another, take that mutants, make another, give them back to the positive charge residue. So here the trick is, it's not the exact same amino acid, but the charge is maintained positive. And now you see a little bit, you see they regain their internalization activity. So the summary for this part, the positive charge of residue of PlyC-B does mediate the internalization to give this enzyme go intracellular, penetrating into the epithelial cells. And then back to the next question, how does this enzyme go intracellular? Some of you may be familiar with one phage enzyme called choline that can have the ability to penetrate the membrane. But the choline, they actually punch a hole in the membrane. We wonder if that's the same scenario here. So we did a membrane integrity test in our epithelial cells. The idea is if the membrane was damaged, the dye can be penetrating through the membrane and light up the cells. So first we treat our epithelial cells with PlyC, and then we add our dye, and if nothing staying there. Then we have a positive control, is we can permeabilize the epithelial cell first, and then add the membrane dye, because the membrane is compromised, and then you see the light up. So indicating the way the PlyC, the enzyme go intracellular, it doesn't punch a hole there. And then we want to investigate what's the mechanism of the PlyC to go intracellular. As I showed you before, this is a cell-penetrating peptide. There are some well-characterized endocytic pathway for allow them to go intracellular signal. Here we decide to take a different inhibitor that specifically inhibit the different endocytic pathway, like clathrin-mediated endocytosis, caveoli, or micropenocytosis. But long story short, none of the inhibitor we applied there actually can inhibit the PlyC internalization, but there's one, it's called the word Manning, that actually can inhibit the whole process. And this is a PI3 kinase inhibitor, which indicating this could be using a PI3 kinase dependent pathway. Of note, the Ward Manning actually as also inhibit the group A strep, the bacterial internalization, give us suggestion maybe they share the same the pathway. Just to give you an idea how this Ward Manning inhibit the trafficking, intracellular trafficking of the PlyC. So again, we use confocal microscopy. So what you're seeing here is two epithelial cells adjacent to each other, and you have a nuclear stance Debbie, and also you have a cytoskeleton staining to establish the boundary. And the bright field showing when you treat with Ward Manning, you see this bubble or vesicle look-like structure, the arrow point over here. And then you add a fluorescence label PlyC, the Endolysin, you see they have a sort of a localization too. And when you merge those two pictures together, and you see those PlyC localization actually superimposing, overlapping those vesicle, suggesting the trafficking of PlyC actually be trapped or blocked by this inhibitor in some type of vesicle formation. And the last part we want to say is we're always talking about intracellular. What is the intracellular environment? What we did is we want to ask where does PlyC go in intracellular signaling? So in cell biology, you have a bunch of subcellular marker you can stain your cells. We use two of them, the early endosome and the lysosome. The reason why we use them because most protein uptake pathway, the protein go through fuse the early endosome, eventually go to the lysosome. And here, sure enough, when we actually apply, you have fluorescence label PlyC showing here in red, and early endosome stain as green. So here you see there's not a lot of co-localization, only 3%. But when you actually see the lysosome, again, showing the lysosome stain as a green, and also the red dots represent our PlyC, the Endolysine. You see a majority of PlyC actually predominantly co-localized with the early lysosome, which indicating that they're utilizing lysosome-dependent compartment pathway. So to quick summary about the part I just mentioned, the viability test showing the PlyC internalization doesn't compromise the membrane integrity, and also the intracellular PlyC trafficking can be inhibited by a PI3K kinase inhibitor. The intracellular PlyC is predominantly localizing the lysosome, which is consistent with some previous typical protein uptake pathway. To finish up the take-home message here, we found one analyzing the inherent ability to go intracellular and remain functional in the intracellular environment. And we found the positive charge on the surface actually mediate this process. How can we learn about this PlyC story? We showed this symptom is very unique on PlyC, but the rational theory is if you have a super positive charge surface, that could be actually enhance your ability to go intracellular to make your antimicrobial, like Endolysine, could be a future therapeutic potential. With that being said, I would like to thank the lab person in our lab, also Dave Donovan sitting there for providing the B30 Endolysine, and also for the funding resources. The last part is I want to give a special thank to my boss. My boss happened to be here. He's trapped in the airport. But what interesting, most of you may know what incredible personality he has. And the way he actually trained us to think as a scientist is every time when you have a result or data, he always say, don't bring me just a problem, bring me a solution. So this is really help us as a student to think how you can approach that, how to think as a scientist. With that being said, I'll stop there and would like to take any question. Thank you so much.
So David Christensen from NCBI.
Yep.
So it's a good thing I'm not in his lab because I have a problem but no solution. So I was wanting to ask, I feel like you did a good job showing why this endolysin could cross the membrane and go inside the cell.
Mhm.
What about the other proteins that you showed early on that were TAT labeled? Why couldn't they cross? So this relates to your last future direction, how to go forward to other proteins.
Yep. Great point. Thank you, Dave. So the reason why we did a lot more investigation why those TAT label endolysis doesn't go intracellular, which is not consistent with other previous literature. So one thing is the way we look at whether they work or not is based on co-culture assay. We actually look at the recovered bacteria. So we think the reason why there's one possible reason they probably don't go intracellular. And that's actually confirmed by our microscopy work. So when you fluorescent label those endolysin, like B30, they're trapped for some reason, they're trapped on the membrane. They have very small portion of the label protein actually go intracellular, but we think that small portion is not enough to cure intracellular pathogen. And also, our PlyC is a very active, very broad pH spectrum, which especially with lysosome is very low pH. Our PlyC is still active. And the B31, the other endolysin, they're not active at low pH environment. So to answer your question, whether they doesn't go intracellular or they go intracellular but not that active, we're still working on the mechanism. But for sure, we're going to be able to know that by using the confocal microscopy to see whether they go intracellular or not. Yep.
Thank you. One more question?
Mark.
So the question is, where is Streptococci in the epithelial cells? Is it in the lysosome also? Or
Yeah, so the way the strep go intracellular, they're well-characterized pathway. The strep was uptaken by the epithelial cell by phagocytosis. They form a phagosome compartment, and eventually they're going to fuse to the lysosome, which the colocalization we saw. So there's some reports saying the strep actually go lysosome as well. Yep. Alrighty.